The last speaker, Hank Greely, is at Stanford University Law School, very active in the bioethics program out there as well. I think this is his 18th trip to the East in the fall. And we asked him to give us a look at future possibilities of governance. So take it away.
Thank you, Art. So I'm going to start this talk by doing something I never do. And those of you in the audience who know me know I'm not going to use slides. Instead, I want to start it by thanking the organizers for inviting me to talk on this. I never do that. It's not because I'm not grateful. I'm a ham. I'm grateful for any audience and any chance to have an audience. But I'm really grateful for a chance to talk about this topic now. I think this topic is perhaps the most important policy topic facing the biosciences today, and it comes a week after I spent most of a week at the National Academy's other building at the great Human Gene Editing Summit of 2015, listening to 2.5 days focused entirely, almost entirely, on humans. I think human gene editing is of some interest. I think it is much less immediate, much less imminent, and much less important in the long run than editing non-human organisms. So I'm going to cheat and breach my instructions. I'm not going to talk about issues with research animals. I want to talk about animals and non-animals, gene editing in all sorts of organisms, and I want to talk about them for research uses and for non-research uses. I think that makes sense because, first, many of the things I'm going to talk about will go through a research phase. Secondly, regulations affecting the broader system may well affect research as well. And third, public reactions and blowback from the non-research uses may well have effects with respect to research animals. There are lots of possible non-research uses out there. Just last week, Alta Charo and I published a little piece in the American Journal on Bioethics with what we thought was the clever name CRISPR critters and CRISPR cracks, about the different kinds of CRISPRed organisms and the different kinds of regulatory gaps they might expose that we would see. I'll give you 12 different possible uses, limited only by my imagination. We've talked a lot about research animal creation using CRISPR and other gene editing. There are also issues of human health, whether it is farm animals with the pH, whether it is mosquitoes to prevent the transmission of malaria, whether it's xenotransplant, genome-edited pigs. All those are human health uses. There are agricultural uses. There are biofuel uses. There are bioremediation uses, using genetically engineered microbes to clean up waste sites. There are also interesting questions around de-extinction, an increasingly interesting theme. And what Alta pointed out to me, domestic de-extinction, bringing back old varieties of domesticated crops, for example. There are interesting questions around gardening. The blue rose, long sought by breeders, may finally become a reality. There are questions around pets. I mean, look what we did to poor wolves in the course of 10,000 years. And with CRISPR, we might even be able to do it with cats. Imagine a Great Dane-sized cat. It's a frightening thought. There are the possibilities of exhibit animals. Don't make woolly mammoths to put them back in the environment. Make them to put them in a zoo, or to sell views of them, or make a dragon, or make a unicorn, or make anything you think you can make money off of. And then there are questions of, there's the possible uses on the dark side for bioterror and biowarfare, which don't get talked about very much, but are a very real potential use of CRISPR to modify organisms to be agents, pathological agents, either aimed at humans or aimed at the crops and the livestock upon which humans depend. So there are a lot of potential uses out there. I want to talk about how we might deal with them, and really I want to talk about three things. First, what should we be worried about? What might we be worried about? Second, how can we try to deal and approach some of those problems? And third, how can we move the system to a position where it will approach rationally some of those concerns? I do want to note, I don't say this in terms of as an opponent of genetic engineering. I think it is a spectacular technology that is likely to do great good for humans and other organisms on this planet. But like all technologies, it can be used well and it can be used poorly. It can be used prudently or it can be used negligently. It can be used for good purposes or it can be used for evil purposes. And we need some sort of thoughtful system of governance, broader than regulation, to help us try to maximize the benefits from this technology and minimize its harms. So, first, what should we be worried about? What might we be worried about that would require some sort of governance or regulation? 5 things. First one, welfare. And this, I have to admit, is mainly animal welfare. I've been trying to think about microbial welfare or plant welfare. Sometimes I think my plants scream at me when I don't water them enough, but I think maybe I'm being oversensitive. I think the welfare issues are largely animal and animal over a certain level. I won't say higher or lower, but differently animaled. And they deserve real consideration as we make new organisms, as we modify organisms to make new ones. What are the implications in terms of the quality of life for the organisms we've made? What are the implications for the mothers or other progenitors of those organisms in terms of quality of life? Those welfare issues demand attention. Second, and I think this is probably the biggest one, what are the implications of release of those organisms for the environment and for health? The environmental implications we see kind of a preview of whenever we think about exotic species. Invasive seems so laden, but species coming from other areas that end up significantly disrupting an environment, whether it's kudzu or the Chinese mussels or other things that come in, mess up an environment they're in. The possibilities of gene-edited organisms having and doing environmental damage is non-trivial. There's also the possibility of health harms. They may be not only direct health threats or safety threats, but they may turn out to be vectors for other kinds of diseases. And we don't have to look farther than the United States in the importation or the successful establishment of breeding populations of certain non-native mosquitoes, bringing with them certain non-native diseases to Southern California, to Florida, to Hawaii. The possibilities of health implications are non-trivial. Third set of issues. This is kind of hard to give a good example of. I've got one, I'm not sure how good it is. Genome-edited organisms or their creation creating dangerous knowledge. The only example I've really got that I think makes some sense is the story from a few years ago about gain of function with respect to the influenza virus, which were, after all, genome-edited organisms, if you count viruses as organisms, which I think is sort of an iffy issue, but genome edited to become more infectious. And that knowledge, if it got out, it was argued would be dangerous. And there was about a year-long hoo-ha at the federal government level and with Science and Nature about whether certain papers would be published. Genome editing may provoke knowledge, which if published widely on the internet would make us all nervous. Fourth thing to worry about, malice. Actual people trying to use genome editing to do harm in terms of biological warfare, in terms of crop warfare, in terms of targeted warfare. If you had a CRISPR construct that somehow did something terrible to disrupt a gene, and its focus is on genes that provide adult lactose tolerance, that hits some parts of the world's population much more heavily than other parts of the world's population. It's not perfect. You'll never have a weapon that wipes out the South Irish and leaves the North Irish fine. But some sort of targeting becomes possible with CRISPR. That's a concern. Malice, the ability to do this, when people say 3-year-olds can do it, and I pushed a little on one of the people who said 3-year-olds could do it, and said, well, you might need college students in order to actually get them into organisms. 23-year-olds can do it. A lot of 23-year-olds out there. Some of them have biological labs. Many of them have garages. ISIS has, for the past almost year, controlled Mosul, which is the home of the second largest university in Mesopotamia, which has a medical school. And last week they executed someone, the chair of the physics department, because he refused to help them build biological weapons. So the biowarfare potential for genome-edited organisms is significant. And the last is the hardest for me to kind of grapple with, but I think we do have to grapple with it. I'm not sure, I'll come back to this, what we should do about it. But there are a host of concerns about this that I'm going to label moral. And I'm going to put moral in quotes, but they're intangible concerns. They're things that bother people. I think some of them have to do with playing God, or in a secular version, playing Darwin. You know, if natural selection had wanted this organism to be this way, it would have made it this way, as the secular version of God. Some of them have to do with purity. You know, it's surprising how often people get offended about where a gene put into one organism came from. Somebody mentioned that with the salmon, people keep saying it's an eel-like creature, as if that somehow makes a difference. So there's a purity concern. There's a yuck factor concern. How many of you remember seeing the photo from 18 years ago now of the Vacanti mouse, the mouse with the human-looking ear on its back? How many of you, raise your hands if you remember it. When you first saw it, how many of you said, yuck? How many of you said, cool? How many of you said both? So there is a yuck factor. I was sitting on a plane the other day, as I seem to be doing every other day, and next to me was a guy whose computer had a weird logo on it. It turned out he was from a company called GitHub, which is some computer thing, a world I don't understand. The logo for that is the octocat. It's a cat head, a cat body, on 8 octopus tentacles. As a logo, it's somewhere between creepy and cool. If somebody were actually to make one of those, I think the yuck factor would probably kick in fairly strongly, even for people who don't like cats, or maybe who don't like octopuses, octopi. So those are 5 things we might want to regulate about. What can we do about them? I have 8 suggestions. First really builds on something Art Kaplan said. If we want to take IACUCs as a regulatory body for this, or something IACUC-ish, if that's a word, it's a wonderful thing about English, we can make words up as we go along. We need to think about expanding IACUC's jurisdiction, abilities, and concerns. 2 guys in a garage don't have an IACUC. People making pets don't necessarily have an IACUC. People making plants or microbes don't have IACUCs at all. And people dealing with non-vertebrates, with invertebrates, don't have to go through IACUCs. So if you're thinking about all organisms in all settings, the IACUCs and maybe the Animal Welfare Act, which is the basis for some of the regulation, would need some expansion. Second thing to think about, registration requirements. Registering who's doing this, getting some idea of who's out there and who's doing this. Now, most of you are from academia. This doesn't mean very much to you because the NIH knows who you are and where you are. You are well registered. But again, companies that are doing this, you know, at some point they may need to register with the FDA if they're making an FDA-covered product. But if they're not, they may not. The 2 guys in the garage aren't going to be registered. Knowing who's doing this work is important. The second level of registration, though, is knowing what they're doing. Making sure somebody keeps a record of what changes they're making, what gene constructs or what genes they're disrupting with various experiments. That could get very detailed as people change what they're doing from time to time. But detailed protocols and a requirement that those somehow be kept and be made available would be really useful if later you're trying to figure out what this escaped organism actually is and does, and maybe where it came from. A third possibility, and I don't know whether this is technically possible, though I think there may be some routes to it, labeling. And I don't mean labeling whether the organism is GM or not. I mean actually labeling the organism. I was thinking about this in terms of Craig Venters putting all sorts of stuff in the little microbe he made. He added a bunch of things to the code. Maybe you could require the insertion of a code that said where this came from. And again, that's not going to help you with ISIS, but it may help you figure out when you find an organism in the wild and you don't know where it came from, it may give you some clues as to who did it and where it came from. Fourth, and I think this is really important, and this applies to the research setting as well. It's already there to some extent, but I think it's going to need to be expanded. Worry about preventing accidental release. Most of the research things are well contained. They're not going to get out. At least we hope they're not going to get out. But if they do get out, the potential harms and the potential public harms, loss of trust harms, those sorts of things can be substantial. And of course, there's nothing like that that applies, again, to the two guys. And I'm using guys generically. It could be two women. It could be a guy and a woman. It could be any selection you care about. The two guys in the garage. So some sort of work to keep accidental release from happening. Fifth, figure out how to regulate release. We have some of that now with USDA, sort of with FDA, sort of with EPA. Whether it's comprehensive or not, my guess is it isn't. We've seen USDA turn down jurisdiction of some things that seem like they would be GM. But whatever it is, we need some system that says this is ready to be released. Somebody has looked at it and worried about the ecological effects and the potential health effects. Now, who would do the looking and what kind of studies you would need? How much of sort of controlled release, caged release studies before you do what kind of open release studies? I was really interested to hear Carolyn Plunkett's discussion of what had been done with respect to the diamondback moth, those kinds of studies. What you would need would vary from circumstance to circumstance, and who would do the regulation would be a really interesting question. But we shouldn't put genomically edited organisms into the environment without giving some thought to whether or not it's safe. Sixth issue, remediation. So we should give some thought before something's released into what we would do if it went wrong. Is there a plan to kill the suckers off in some useful and productive and easy way? Is there a plan with gene drive, the possibility of actually changing the genome of every individual in a species on the face and under the face of the Earth is realistic? Is there a plan to reestablish the old species? If the new one turns out to be a bad idea, or the old version, have we stored that DNA? Can we fix it if we break it? Should people, before releasing, be bonded so that we know that they can pay for the sorts of damages there would be, bonded or insured? Those kinds of regulations, looking forward to the fact that mistakes will happen. How do we clean them up? How do we clean the mess up? Good idea. Seventh, on the national security stuff, on the biosecurity stuff, it's hard to come up with real good ideas for that. And increasing surveillance, I suppose. I don't think we can realistically monitor and control all of the reagents sold for CRISPR everywhere, all of the guide RNAs sold anywhere. Be useful to have the people providing guide RNAs or RNA to pay some attention to the sequences that they're providing and what those uses might be. But that's going to be hard to second guess. We'll have to try. But frankly, I'm not sure I see any good way forward other than human intelligence, by which I mean having people involved in the terrorist groups and the other groups to try to tell us what they're doing and what they're thinking, as well as maybe electronic intelligence and so on. I think this is going to be a genuinely hard problem. We also may want to revisit the question of some restraints on publication. The NSABB, the National SABB, Science Advisory B Board, Biological Board, thank you. You know, I can never get that one straight. Basically tore itself apart over that, and those who dissented then weren't reappointed in quite a purge. But the issue hasn't gone, and there are First Amendment questions as well as principled questions. That's a hard question. I don't know the answer to it, but I think we may need to think about it. And then last, should we have some moral test, some moral check on what can be made and what can be released? Should there be some body that says, well, yes, you could make an octocat, but you shouldn't? And whether you shouldn't because it's impure, or because it's unnatural, or because it's against God or Darwin's will, or just because it's going to provoke such a public yuck factor that all of science will be harmed. At least all of biological science will be harmed and discredited, and funding for NIH will go down, and scientists will be thrown out of work, and all those other things. Whatever the reason, to the extent there is going to be a public reaction, it is worth thinking about whether we want to regulate it. How would you regulate it? Would it be at an independent IACUC level? Would it be at a federal RAC level? Beats me, but we have to think about whether we want regulation for that or not. Now, some problems. First, how do you implement it against the two guys in a garage? Really hard. High levels of criminal fines for not registering might do something for it. I'm not very worried about universities or major industries or research institutes. I am more worried about do-it-yourselfers. Biohacker facilities have grown up all over the country. Somebody just raised $25,000 on Indiegogo in 3 days to produce do-it-yourself CRISPR kits. He plans to send out a kit to modify yeast and a kit to modify bacteria and send them to anybody who's interested so they can learn how to participate in this revolution. Maybe we should track everybody who gets a do-it-yourself CRISPR kit. But doing the implementation is going to require some very difficult, it's going to be very hard and require some very creative thinking. The institutions, not such a big deal. The individuals, the democratization. Generally, I'm in favor of democracy, but it occasionally has its downsides, as Winston Churchill used to remind us. So I think that's a problem. Another problem is the international scene. Trying to get something uniform done internationally. Trying to get anything done in some countries, which have much bigger and more immediate concerns as far as they can tell, than worrying about genetically modified organisms, is going to be hard. And in my experience, when you get international conventions, you tend to get the least common denominator and things that often aren't enforced anyway, aren't enforceable or aren't enforced. So the international side is going to be another set of problems. Let me suggest two possible sort of offbeat solutions. One of them Andrew pointed out, Andrew Torrance, the tort system or the product liability system. The idea of strict liability might give some people pause if that aqua advantage salmon, despite everything that you've done, does turn out to be horrible. And if it means that you're strictly liable for all the damage it causes and you go out of business, that focuses the mind a bit. So strict liability may be a possible check. Another one might actually be the patent system, because people who own patents control uses, at least in theory. How good that would be, unclear, but it adds another possible enforcing or governing mechanism to the force of the state itself. So finally, where do we go from here? How do we get from these things we need to regulate and ideas about how to regulate, things that we might want to regulate and ideas about how to regulate them, to actual regulation? I am afraid, although I cannot say for sure, I am afraid that the coordinated framework is not going to work, that there are cracks through which CRISPR creatures fall between the FDA, the EPA, and the USDA, that some sort of new legislation is probably necessary. And I say that with great dread, because the idea of things that come out of Congress scares me. I remember a few years ago, the then Senator Brownback's Human Chimera Prohibition Act of 2006, which would have made a number of my friends felons had it passed. Congress can do some screwy things. What Congress mainly does is nothing. Usually Congress acts in two circumstances: when there's a perceived crisis, in which case they hurry up and do a bunch of stuff whether it makes sense or not, or when there's been a long period of consensus building and different interests brought into coordination. Sometimes you can combine those. You can be ready with a bill that has gone through interest coordination, and when a crisis strikes, you offer the bill, they're desperate for anything, they take that. But I think in order to get decent regulation, we need to be thinking about it and building that kind of consensus across stakeholders to come up with what works. And by stakeholders, I mean not just regulators and consumer advocates and non-governmental organizations and people involved in the industry, but people from all over. There is some movement along that line. The National Academies we haven't really talked specifically very much about gene drive, which makes all of this scarier in the context of non-humans. The National Academies has a study on gene drive that's supposed to report sometime, I think, relatively early in 2016. The Office of Science Technology Policy has called for a revision or rethinking of the coordinated framework. I understand there may be a National Academies study report on that, although there's still some uncertainties about funding and timing. Those are good. More is merrier. We need more work in thinking through how we should actually proceed with this. And I do share the sense that we are at a special time. CRISPR-Cas9 is not the first gene editing. We've been doing gene editing for a long time. We've been doing breeding for a long time. One important question, by the way, is why should we worry so much about this when we don't worry so much about exotic plants coming in, about traditional breeding, about breeding enhanced with ionizing radiation, and so on? That's another good question to deal with. But CRISPR-Cas9, I think, marks an inflection point. Somebody mentioned the Model T earlier, but a different way than I want to use it. The Model T was an inflection point. Before the Model T, there were cars. They were expensive. They broke down all the time. They were crummy. You had to hire a chauffeur mechanic in order to be able to use one, and you got 10 miles an hour. Model T came out a little over 100 years ago, democratized the car. Anybody could own it. Anybody could run it. My state of California went from about 2,000 cars to about 25 million cars. And that changed everything. It changed the air we breathe, the cities we live in, how we have sex. All sorts of things changed with the ubiquity of cars. And all Californians are now centaurs, half human, half car. It's not entirely false. So CRISPR is the Model T. We're not going to be using CRISPR-Cas9 in 10 years. We probably won't be using it in 2 years very much. But it's where this technology became democratized, became readily available, became much broader. We need to be ready to cope with the consequences, and we need to start last week. Thank you.